Hello, and thank you for joining us for this discussion about the innovative technology the Cycle for Survival community helps fund. My name is David Solid, and I am a medical oncologist and laboratory scientist. As a member of the genitourinary oncology service, I specialize in treating cancers of the prostate, bladder, kidney, testis, and other related cancers. I've also been a Cycle for Survival participant and supporter for over 10 years, helping lead the charge for our incredible Team HOP, which has raised more than $2.3 million to support research in MSK's Human Oncology and Pathogenesis program. Each season, the Cycle for Survival community takes on a month-long challenge to raise funds for innovative technology that helps our scientists accelerate discoveries. I've been fortunate enough to have been a recipient of this funding, and so I know firsthand just how crucial this type of support is to our work. I'm excited to be joined today by three of my MSK colleagues who share this privilege: Kat, Donna, and Sarab. Thank you so much for taking the time to be part of this discussion. To kick things off, I'd love if each of you can introduce yourselves and tell us a bit more about what you do here at MSK.
Hi, my name's Katja Gentanakis, and I'm the chair of the Developmental Biology program. Developmental biologists are interested in how cells regulate their identity and how together they build tissues and organs during embryonic development. The premise being that cancer cells will oftentimes reenact developmental programs out of context. Furthermore, only with a knowledge of how tissues and organs are built will we be able to aspire to our future goal to repair, replace, or regenerate damaged or diseased tissues.
Hello, everyone. I'm Donna Peer, chair of the Computational and Systems Biology program. Myself and my amazing colleagues and additional faculty members in the program are originally trained in mathematics and computer science, but have become bilingual, understanding both biology and computation, and are able to develop novel, powerful computational approaches to interpret data to bring new understanding to biology and medicine. I also direct the Gerian metastasis and tumor ecosystems, which brings cutting edge single cell technologies to puzzle out cancer one cell at a time.
Hello, my name is Sarab Shah. I'm the Chief of Computational Oncology at MSK. And computational oncology is a field where data and computing meet cancer research and treatment. And so we use computation, machine learning, artificial intelligence technologies to take data derived from patients and understand that data so that we can best provide treatment solutions for those patients. And overall, we do research in that area to develop new computational methods that can integrate different data forms to derive at that solution most effectively, and also to better understand the biology of tumors and how they evolve within the host of the patient.
My team and I have benefited from these funding challenges in the past, such as the December Challenge, which in prior years raised money for a DNA sequencer, a liquid biopsy system, and genetic profiling, all of which was technology used to support the Center for Molecular Oncology at MSK. I also work on the MSK Impact and the Make an Impact initiative, which provide DNA sequencing to some people at no cost. Cycle for Survival funding has played a key role in the launch and continued use of Make an Impact as a groundbreaking diagnostic and research tool. Since we launched the MSK Impact program in 2014, we've now sequenced more than 70,000 tumors, and this has had a direct impact on how we now treat our patients. As I know, some of our panelists have also received funding from a December Challenge or March funding challenge through the Cycle for Survival community. This leads me to my first question. Kat, in the 2020 season, the Cycle for Survival community raised over $1 million for a light sheet fluorescent microscope. Can you tell me a bit more about this technology and its impact on your laboratory research program?
Okay, thanks, David, for the opportunity. My colleagues and I are thrilled that the Cycle for Survival community has allowed us to bring in an innovative new microscopy or imaging technology to MSK. And I should say that we didn't just buy one microscope, but the funding, the support that we received allowed us to buy two microscopes. These microscopes allow us to visualize molecules in cells and cells in tissues at unprecedented resolution. We can investigate specimens in 3 dimensions and also, in many cases, over time in live specimens. So, what we're very interested in doing is looking at dynamic movements of cells in tissues to ask how cells move to different locations, for example, during metastasis. And also we're interested in investigating how neighboring cells and tissues communicate and ultimately build organs normally or lead to disease progression in a patient.
Great. Don and Sarab, in March of this year, Cycle for Survival supporters raised $1 million to help purchase additional computing equipment for MSK computational oncologists. Can you each tell me how this equipment purchase and how these resources are really helping to drive innovation and improve cancer care for our patients?
These amazing new single cell technologies, such as the light sheet microscope that Kat described, as well as single cell genomics, which allows us to view the activity of the entire genome, tens of thousands of genes in tens of thousands of cells, one cell at a time, really, again, allow us to look at cancer cells and the interactions with their environment at unprecedented resolution and scale. We can measure the individual, nasty, rare, malignant metastasis initiating cells and really observe and figure out how that individual cell can trick its healthy environment to support it rather than search and destroy it. These technologies are really particularly potent for the rare cancers because now we can take an individual cell, profile tens of thousands of cells from that individual's cancer, and understand that rare cancer, that individual patient, and the vulnerabilities of that patient's cancer to allow us to think about potential therapeutic approaches to help the patient. We have the data. It's incredibly rich, yet enormously complex. We're talking about billions and billions of data points stored over terabytes of data. Therefore, it's absolutely critical to have powerful additional computational power to interpret this data. And this generous gift by the Cycle for Survival community has really enabled our work.
Exciting. Sarab, can you build on that? MSK's computational oncology program is home to one of the largest data sets anywhere. Can you tell us more about how this program and how Cycle for Survival is powering it?
Yes, indeed. So I'd also like to just thank the Cycle for Survival community for providing this incredible gift. Uh, One of the things that we execute in computational oncology is the analysis of the genomes of individual patients, as has been mentioned before. And this allows us to match effective treatments to individual patients so that they have a better chance of responding to that treatment. And this is a field called precision medicine. The execution of this over a number of years has accumulated a vast resource of data, sequencing data from tumors from patients at MSK. And this allows us to study rare cancers because there are enough of those examples that we can start to recognize patterns in the genomes of those tumors to better understand their cancer biology and how they might respond to potential treatment options for them. Furthermore, we're now starting to layer on additional information onto the genome, such as medical imaging information, single cell analysis, as Donna eloquently spoke about. And in order to do this, we use machine learning technologies and artificial intelligence techniques to integrate these data sets together to better understand them as a holistic package. And that will allow us to improve precision medicine and take it to its next generation.
So it's really exciting technology. The science is spectacular, but I'd love to hear from each of you about how these technologies are leading to advances that are directly helping our patients. Kat, can I start with you?
Absolutely. These so-called light sheet confocal microscopes that we've now brought to MSK are allowing us to visualize and contrast events as they take place in normal versus diseased tissues and organs. So there's no reason why over the next couple of years we can't start to visualize cells in rare cancer samples. I think one of the bottlenecks that we're currently facing is that really these types of microscopes that we're now sort of establishing and starting to use at MSK are generating, as Dana alluded to, huge data sets. Any one data set is terabytes in size. And what we're really relying on are computational advances that will allow us to work with the data, to query the data, because really we're now at a crossroads where we're generating data of unprecedented resolution, size, and detail. And we really have to be equipped computationally to analyze these types of data.
Great. So, Donna, how do you see single cell technology directly impacting patient care? Is it identifying new drug targets, or is it telling us who's going to respond and who's going to be resistant? Can you give us a little more insight about how this is impacting our patients?
I want to echo Sarab and repeat the word personalized medicine. And really, these single cell technologies and all the data that we've been talking about really make the promise of precision medicine and personalized medicine possible. Because for each individual patient, we could look at all these different data modalities, be it the DNA, the clinical modalities such as PET and CT, as well as single cell genomics and high resolution microscopy in individual patients to understand that individual patient's cancer, the vulnerabilities of that individual's cancer, and what therapies are most likely to affect it. Our bag of potential therapies is still not as rich as it needs to be, and the real power is future therapies and better solutions for patients of the future. And I can share one exciting story. Our single cell technologies really were able to discover a new surprising vulnerability in leptomeningeal metastases. These are really terrible metastases. And I'm really proud to say that this week, the first patient is undergoing a clinical trial for a therapy that our computational approaches discovered. And there are 5 more clinical trials that are in the works, derived from direct predictions of our computational approaches applied to data using the powerful computation that is so generously provided for us by the Cycle for Survival community.
So, Rob, we hear these terms like big data, machine learning. How are these starting to impact our patients? How is this large scale data set and computational resource really transforming the care of cancer patients?
The most effective type of machine learning and artificial intelligence leads to understanding. And I think this is where we've made some advances now by taking these incredibly large and detailed data sets that have been referenced by Kat and Donna and using these techniques to understand, for example, how does a patient's immune system recognize a cancer that's developing and forming? And furthermore, how does a cancer cell then actually evade and hide from that immune system? And what we're learning is that different tumors do this in different ways. And this has a very powerful implication, even within a single disease like a type of ovarian cancer that we study. There are at least 3 different ways in which the cancer cells hide from those immune cells that are trying to survey them in the patient. So this has been a powerful discovery through using artificial intelligence and machine learning technologies on these huge data sets to recognize these patterns. And this allows us to then develop further therapeutic strategies for those patients and tailor those strategies in a new way that goes beyond just the genome, which has been incredibly effective, but now we're taking it to the next level and layering on additional information.
Yeah, I mean that's very similar to what we've seen in our work as well. We've studied BRAF for a long time, which is common in many different cancer types, but we find that patients with BRAF mutations don't all respond the same, and it actually sometimes ties back to what Kat's working on, that there's different developmental programs in these cell types that actually condition how they respond to BRAF inhibitors. So my hope is we can bring all this science together here at the institution to really drive innovation for our patients over the next few years. Well, that's all the time that we have. I want to thank our panelists for sharing their insights. Your support of initiatives like the December Challenge is what drives innovation and accelerates progress in rare cancer research and treatment. Join us after the program for a live Q&A. We would love to hear from you and hear your questions. Thanks again to our panel and each of you for tuning in. Take care, and I hope to see you all on the bikes next spring. Let's ride.